We begin our journey at the PCI Creative Science Park Aveiro region in the laboratories of Meta Tissue, where cutting-edge biomaterials are developed for clinical and research applications. Here, the process of hPLMA production is carried out under strict quality and safety standards. The platelet lysates are stirred in a bioreactor equipped with a pH electrode. Methacrylic anhydride is carefully added, allowing the reaction to proceed under controlled conditions. Following the reaction, the material undergoes dialysis to remove any unreacted methacrylic anhydride. This step ensures purity and safety of the final biomaterial. Next, the product is sterilized by filtration and lyophilized, yielding a stable powder, the final hPLMA. Each batch is required to meet critical quality standards, starting with its ability to form hydrogels. To assess this, the precursor solution is dispensed into a well on a glass slide and exposed to a specific wavelength of light, initiating photopolymerization. The resulting cross-linked network confirms the material's ability to form stable hydrogels under controlled conditions. To ensure compliance with safety and functional requirements, each batch is tested for sterility, absence of mycoplasma, biological activity, degree of chemical modification, and endotoxin levels. As a final validation step, a cell culture assay is performed to confirm cytocompatibility. Human cells are seeded within the gel matrix, and their viability is assessed through fluorescent staining. Live cells appear green, while dead cells are marked in red, allowing quantitative analysis of cellular response within the hydrogel environment. After successful completion of all quality assessments, the batch is formally released for use. A certificate of analysis is issued to confirm compliance with all critical specifications, including safety, functionality, and performance. Once the hPLMA is ready and fully certified, the workflow continues at the University of Aveiro. Here at DEMAC, the Department of Materials and Ceramic Engineering, the PL bioceramic ink is formulated by combining bioactive and structural components. The process starts with the precise weighing of calcium phosphate powders and human platelet lysates. The powder of calcium phosphate is characterized by high purity and defined particle size distribution, which enhance dispersion and contribute to consistent mechanical performance. The components are then dispersed in an aqueous solution enriched with tailored additives, optimizing the ink's rheological properties for printability and stability. The full mixture is homogenized in a planetary centrifugal mixer at 800 RPM for 2 minutes, ensuring a uniform distribution of ceramic and biological elements. Now that the bioceramic ink is ready, researchers initiate the printing of the scaffold's foundational layer, tailored to foster bone growth thanks to its osteoconductive composition. The bioceramic ink is loaded into a 3 ml syringe equipped with a 410 micrometer conical nozzle. This is mounted on the robocasting system. The scaffold's architectural layout, optimized by eNegi through advanced simulations, is designed to enhance mechanical performance and ensure structural reliability. This design, developed in RobCAD software, is then used to guide the printing process. Printing is carried out at a steady speed of 10 mm per second under ambient temperature and high humidity. A layer-by-layer deposition strategy is used to fabricate scaffolds with controlled porosity and geometry. After printing, scaffolds are placed in a conditioning chamber set to 37 degrees Celsius and controlled humidity, where they harden and stabilize structurally. The journey now continues at the University of Aveiro in the Biomaterials Science and Engineering Lab of the Compass Group, where a new ink has been developed to serve as a structural support for soft tissues such as cartilage. To reach this, the next step is the preparation of PLDN ink, a double network bioink composed of platelet-based elements and crosslinking agents. The formulation starts with the weighing of hPL, hPLMA, the LAP photoinitiator, and the crosslinkers EDC and NHS. After being dissolved in PBS, each vial is placed on a vortex mixer, a vibrating platform that facilitates rapid and uniform mixing of the solution. The cross-linking reaction begins with the addition of EDC and NHS to the hPLMA solution. This mixture is incubated at 20 degrees Celsius and 1250 RPM for 5 minutes in a thermomixer. Then a solution of hPL and LAP is added. At this stage, a biocompatible dye is also incorporated into the formulation, allowing the PLDN hydrogel to be visually distinguished from the underlying bioceramic scaffold. An option has also been explored to reinforce the PLDN hydrogel with functionalized hydroxyapatite nanoparticles provided by Kuros. These particles integrate into the network, improving both stability and osteoconductive potential. The full formulation is maintained at 37 degrees Celsius for 3 hours to complete the cross-linking reaction, resulting in a ready-to-use PLDN ink. A polycaprolactone PCL mesh is printed via melt electrowriting at Maastricht University to serve as an interfacial layer between the bone-like base and the cartilage-mimicking top layers. The development and optimization of this microfibrous layer is supported by EnvisionTech, whose 3D bioplotter technology, upgraded within InterLynk, enables multi-material integration and facilitates co-processing with electrospun fibers and complex scaffold architectures. This microstructured mesh features high spatial resolution and controlled fiber orientation, enabling a gradual mechanical transition between the rigid ceramic scaffold and the soft hydrogel layers above. It also provides a favorable topography for cell attachment and guides tissue organization across the bone-cartilage interface. Now we're back with the Compass Group at the University of Aveiro, where it is time to create the upper soft tissue mimicking layer. The PLDN ink is loaded into a 3 milliliter syringe and fitted with a 27-gauge metallic needle. The ink is precisely deposited onto the PCL mesh to form the scaffold to support the regeneration of cartilage tissue. The resulting construct is a hierarchical biomimetic scaffold capable of supporting regeneration at complex tissue interfaces. Once sterilized, the scaffold undergoes preclinical evaluation at Charité, where its biocompatibility, safety, and regenerative potential are assessed through biomechanical testing in clinically relevant models. This integrated workflow, from raw lysates to printed scaffolds, reflects the convergence of biology, chemistry, and engineering. Each step is designed to ensure reproducibility, biocompatibility, and translational potential, contributing to the development of clinically relevant solutions in regenerative medicine.